Okay, hello everyone. Welcome to our first speaker session today. We have the great privilege of having Sebastian Brunemeier with us. I'll just, before we get into this, as people are coming into the room, I just want to put out sort of housekeeping rules. So number one, if you haven't done your onboarding yet, please do that. Just fill it out. You should have gotten an email with a link to onboard and do the onboarding survey for ODLB. We need that because we can't put you in a mastermind group and we can't do curated connections. And most importantly, we can't send you your cool ODLB swag if you don't finish the onboarding. So please do that. Okay, great. So as people are coming in, I'll just briefly get on another note for today's talk. So today's talk is marked confidential, so please do not share the recording or content of this talk outside of our ODLB community. Thank you, appreciate that. And yeah, so let's sort of begin here. Okay. Today we have the privilege of having one of our founding fellows, Sebastian Brunemeier, joining us today for our first speaker session at ODLB. Super excited. Just briefly about Sebastian. Sebastian is one of the brightest and most prolific minds in longevity biotech VC. He was formerly a principal at Apollo Health Ventures, one of the biggest longevity VC firms in the world. He's co-founder of Samsara Therapeutics, co-founder of Cambrian Biopharma, CEO of Cyclone Therapeutics, and advisor to many other biotech startups. He is currently the CEO and co-founder of Immuneh. And just on a personal note, Sebastian is a great friend of mine. He's also been just an incredible force and lending his support for this program. He's, helped out with connecting us to some other great speakers and just in general, giving great advice for running this community. So just want to thank him for that. We couldn't have done it without him. And yeah, so for the agenda for today's talk, just quickly, we're going to have Sebastian share with us some of his thoughts on doing scientific diligence for longevity biotech. So we'll be speaking for 45 minutes, and then we'll have audience Q&A for 15 minutes afterwards. So if you have any questions, think about them, and yeah, we'll put them in the chat, or you can come off mute. So I guess, yeah, without further ado, thank you for coming today, Sebastian. It's great to see you again.
Thanks, Nathan. Likewise, you're too kind. You're the true rock star here, bringing together this incredible group. So looking forward to meeting everybody. So yeah, I'll tell you guys about my approach to technical diligence in the LongBio space. LongBio is a term we're hoping to coin, so tell your friends. I think it'll increase the sort of investor awareness that this is an investable theme if there's a specific shorthand term for it. I should also say a bit of housekeeping. I just came down with a cold the other day. It's not COVID, I tested it, but if I sound congested, that's why, and maybe a bit low energy, as our esteemed President Donald Trump said of Jeb Bush, low energy. So bear with me on that front. China, I own a Gucci store in China, believe me. So I just wanted you all to see my Trump impression. All right, so let's carry on. So the overview, I'll tell you a bit about myself, my own background. LongBio is an investment theme. Big Pharma is leaving R&D and biotech has to step in to fill the gap. The DISCO model of drug discovery that I'm quite partial to. Biorevolutions comparing Geroscience to the dawn of antibiotics. Spending a fair amount of time on venture diligence and new co-formation, which I think is quite relevant to most of you. And then some closing thoughts and sermonizing, if you will allow me. So my own background, I'm a biomedical scientist by training, dual masters in molecular neuro and biotech business management in Amsterdam. Then I was in the PhD program at Oxford that I paused to do biotech, and I've had a handful of other academic affiliations as well. I moved in the biotech space with Apollo Ventures, building companies focused on aging, including Samsara Therapeutics, which is one of the first autophagy enhancing small molecule companies in Oxford. And then I was recruited to co-found Cambrian Biopharma, which is a diversified portfolio approach, a DISCO model, if you will, to aging. And then most recently, I have co-founded Immuneh Pharma, which is still in stealth mode, but or developing small molecules for immune system rejuvenation. And we'll get into that a little bit. I also advise a handful of other groups, including Nathan's new fund, Healthspan Capital, which has a new logo. So just as a bit of an overview for those who are new to the space, this is what I call a wiring diagram of the biomedical economy. So it starts with the public giving money in the form of taxes to the NIH, which goes to universities and research institutes, as well as the public giving money into the public markets and private markets through your, for example, your pension fund or insurance fund or sovereign wealth fund or university endowment. These are large pools of capital that are limited partners that invest in venture funds, but they also invest in public markets. And so sort of at the center is venture capital and biotech startups. As we launch companies in the space, increasingly many venture funds are doing company formation. So it's helpful to be affiliated with an existing venture fund as an entrepreneur in residence or the like, in order to have that sort of aegis, that existing sort of jumping off point. But in a sense, the earliest customer here is big pharma. So the biotech startups rarely take a molecule through phase 2 and 3 themselves. Usually it's in partnership with big pharma, and then they're acquired at some point, or they IPO first and then are acquired. So, but big pharma also has its own ecosystem to deal with. So big pharma actually mostly doesn't do their own internal R&D, preclinical or clinical stage. They outsource it to CROs, like Quintiles or IQVIA, it's now called. The regulators review the data coming out of the CROs, and then the payers also review the data. So as we're seeing with Aduhelm, the Alzheimer's drug that doesn't really work, FDA approved it despite the protestations of their AdCom, but the payers are putting their foot down saying this isn't worth it, to their credit. And then ultimately you have the healthcare providers, the doctors who actually prescribe the medicines. So I think we're in the halcyon years of LongBio, the right time and place. So what are the factors underlying it? Well, we're in a mania, clearly. It may be sustainable like the tech mania was. So after the 2001 popping of the tech bubble, tech has remained a big part of the economy. There's a lot of venture and public market appetite, and there's a lot of capital. There's a lot of savings globally because of demographic aging coupled with low interest rates. So that capital needs to find a home. We also have increased demand for new medicines because of demographic aging. You guys are familiar with the silver tsunami. If we don't do something about the silver tsunami, governments are going to be even more bankrupt than they already are, particularly with respect to diabetes in the Western world. Pharma is not really doing their own internal R&D, so we've got to step in. It's to their credit that they recognize that, because they're just so bureaucratic. There's also an increasingly high bar for an approvable therapy. So the regulators and the public are no longer satisfied with mere symptom palliation. The low-hanging fruit has been picked. So now there's an incentive to go higher upstream in the cascade of damage that causes aging. And of course, as you guys all know, there have been a lot of breakthroughs in the last 20 years in the field of geroscience. And we have a new model for drug discovery, which is the virtual biotech model, which is capital light. You don't need to build up your own lab anymore, as you guys well know. Plus advances in academic drug discovery in universities and TTOs able to spin out technology. So you got to ride the wave. You look at the biotech index outperforming the S&P, which is itself in a bubble because of central bank policy. We're in an even bigger bubble in biotech. IPO valuations are skyrocketing. So if you can take a company public, give it a shot. Capital is flooding in privately as well. So we're seeing somewhere around 23 billion in 2020 going in, which is just incredible. And you see the same thing going on across the board, depending on the stage of the financing. But despite all of this capital coming into biotech, there's really very little going into LongBio specifically. I'm guessing somewhere around 50 to 100 million per year, excluding, a couple of these big names that are not really longevity, but, they're doing public market or SPAC listings and raising capital there. But no true longevity company has done that recently, other than maybe Unity. So big pharma has historically been asleep at the wheel. So according to the former president of R&D at Roche, the biotech industry is increasingly taking up what the pharma industry is neglecting, carrying out future-directed research, thereby creating the conditions for the discovery of new classes of medicines that attack more directly the root causes of disease. He actually said that in the, around the year 2000. So it's even more true today. And David Baltimore, who you guys will know, said regarding the biotech revolution in the 90s, the pharma industry was asleep at the wheel, unable to understand the profound opportunities provided by molecular biology and therefore unable to take advantage of them. And I submit that the same is occurring with Geroscience and LongBio today. But fortunately, some pharmas are, aware, like Novartis or Regeneron seem to be in AbbVie. So pharma's R&D has been declining. So their internal rate of return is less than their weighted average cost of capital. Basically, they're losing money on R&D. And it's just going lower and lower because of a lot of reasons, but one is bureaucracy. But there's good news. So there's a shift from internal big pharma R&D to the decentralized biotech sector. This data comes from Andrew Lo at MIT and Royalty Pharma. So you can see in the year 2000, the top 30 drugs, only 4 of 30 were from academia and biotech. And those from biotech were from big biotech. Whereas if you come forward to 2018, the ones in blue, the lines in blue here, originated with biotech or academic drug discovery. And a lot of these, by the way, were resisted by pharma. Pharma, for example, Metarex and the CTLA-4 inhibitors. There's a really great documentary about it called Breakthrough about Jim Allison, the Nobel laureate, who is pushing this checkpoint inhibitor therapy that's become all the rage in the last several years. And pharma was ignoring it for about a decade. So just because pharma or investors don't presently appreciate your approach does not mean it is invalid. So don't give up hope. So these days we're seeing about 80% of new drug or the best-selling drugs coming from biotech and academia. The problem is, and everyone on the call hopefully is positioned to help solve this problem, is the valley of death. So going from academic spin-out to or academic project to an actual spin-out. There aren't enough company builders and operators to do that. There's also some very good news out, which is also from Andrew Lowe's group. Probability of clinical success is rising rapidly since, say, 2019 or so. You can see the probability of success from phase 1 all the way to approval, the black line here, has been rising pretty rapidly. And Andrew Lowe argues that it is because of more efficient, lean biotech startups with better incentive structures. But some of that is also due to different modalities like gene therapy, which is more likely to succeed because we know the fundamental underlying biology of the pathology of those diseases. So fortunately, biopharma is not really resisting this trend. They're decentralizing for efficiency. Some data from Bay Bridge Bio finds that for every drug that pharma gets approved, or actually every NME approved per $1 billion. So takes pharma about $2 billion to get a new medical entity approved, whereas startups get about 4.5 approved. So they're significantly more efficient. McKinsey did a similar analysis finding increased NDAs coming from small biotechs. And pharma has a lot of capital sitting on their balance sheet to do deals. So we're really well positioned to do company formation over the next couple of years. So one model to leverage this new theme is called the DISCO. I coined this term a couple of years ago when we were launching Cambrian because there was no term for it previously. And so biopharma business models can be groovy. So what is the DISCO? Well, it's sometimes called a subsidiary portfolio model. You arbitrage the science from the universities into drugs, and you do it in a distributed portfolio approach. So McKinsey came out with an article on this fairly recently, finding a couple of groups outside of the longevity space based on this similar model, raising significant capital. So here are some of the DISCO type diversified models in longevity. You will know Switzerland is well represented, Rejuveron, Maximon are in Switzerland, both rather new, but interesting science going on there. Our friends in the UK, Juvenescence, and then Cambrian in the US and Europe, which I'm partial to. But there are other groups that have paved the way. So BridgeBio, for example, which Andrew Lowe helped found, focused on rare diseases. Roivant, which is sort of mixed reviews, and a handful of others out there. And then you have in the UK, venture capital investment trusts. So these are publicly listed sort of venture firms, similar type of model. So why do a disco? It's an early R&D focus, which pharma is not doing anymore much of. So there's a lot of opportunity there. But it's diversified like pharma, and it's capital light. So you do the key critical experiments with CROs or academic groups. And then that actually is the highest step up in valuation prior to getting in the clinic. The team members have skin in the game. So it's organized into subsidiaries within a large organization. So the operators within that organization have equity. Unlike in pharma, where if you invent a compound, you don't actually have any upside in that compound success. And so this is similar to what pharma tried with centers of excellence, decentralization. GSK tried that. Fortunately, if the project fails, you can come back into the mothership, which incentivizes a clean kill, or what Bruce Booth calls truth-seeking behavior. It's decentralized, hub and spoke model with global reach. So you can arbitrage IP from, say, the European market into the US market, where it's more fairly valued. So moving on to Geroscience. You guys will know much of this, but framing it a little differently. Antibiotics were a revolution in the early 20th century, and they were resisted. Most medical practitioners were very skeptical of the possibility of having a so-called chemotherapeutic that would selectively kill microbes, a magic bullet, because at the time, most people died from infectious diseases. So imagine if you told somebody in the year 1900, okay, we're going to have an antibiotic that, will prevent 90% of the people from dying. That would have been pretty insane proposition. And yet that's exactly what happened. And other age-related diseases took the mantle. So it's a similar paradigm today, which is if you tell somebody, all right, we're going to bring aging under medical control, it seems so fantastic, or at least it did 15 years ago. Now it's actually pretty well accepted with a lot of prominent groups getting into the space. I also want to give a shout out to the development of penicillin. This is an interesting example, a case study. There's a really good documentary on it called Breaking the Mold, about how pharma was asleep at the wheel with antibiotics too, including penicillin. And Howard Florey, who was at my college, Lincoln College at Oxford, who won a Nobel Prize, actually had to force pharma with the help of the US government to manufacture massive amounts of penicillin for World War II, where it saved many millions of lives. And so it's another example of how, pharma is very rarely originating any new revolutionary ideas. It has to come from outside. So you guys will know these pathways. This is not an exhaustive list, but there are a handful of pathways in longevity that only a handful have been targeted. One that I want to give a shout out to is DNA repair. There are very few companies seeking to enhance the fidelity of DNA repair, even though a lot of the supercentenarian data, the SNPs, are gain of function mutations in DNA repair factors, and comparative longevity studies across species finds that longer lived species like the naked mole rat have better DNA repair machinery. And then on the flip side, all of the progerias, all of the accelerated aging diseases are mutations in DNA metabolism factors. So that's quite a clue that we really need to work on DNA repair if we're going to really address aging. And there are very few groups working on it for reasons we can get into in the Q&A. I can speculate. Anyway, so that's a call to arms to seek to enhance DNA repair. There are a bunch of interventions, this is a little outdated, known to genetic and pharmacological, known to extend lifespan. You'll be familiar with a handful of them. And really a lot of what our job is to take these genetic interventions and transform them into drugs. And it's that simple. So you guys know some of these top interventions. I'd like to call attention to heterochronic bone marrow transplant as an interesting one, as well as repair of the leaky gut. Gut barrier permeability is a big issue in aging and chronic inflammation. Hippocrates and Elie Meshnikov both said that death begins in the gut. So I think we should be looking more, not just at the microbiome, which is very hot, but hard to develop a drug around, but actually looking at gut barrier function and the mucosa. All right, so some of the groups in the space, I've mentioned a couple: Apollo, Cambrian, HealthSpan. These are some that I'm affiliated with, but there are a handful of others. Sorry if I left you off the list, but they're proliferating. So when you're fundraising, these are a handful of groups you can get in touch with. For some, I can make an intro for you. And then some of the prominent geroscience companies. So there are some that are publicly listed already. Some of these are only kind of longevity oriented, like Sensei from Cambrian's portfolio, but we think that there's a very large, wide scope for treating many different types of cancer. So it almost qualifies as longevity. And then privately held groups, which you know, these on the right here are some that I've co-founded. And then these are two Apollo portfolio companies. Cambrian has about 10 stealth mode subsidiaries, and Juvenescence has somewhere on that order as well. So keep your eyes peeled for those coming out of stealth. So getting into sort of the bulk of the discussion, how to diligence these assets. This is just how I do it. There's probably a better way, but this is what I've ended up doing over the last few years. So first you have to source the IP. You do preclinical diligence on the science. Most of you are scientists, so this is what we relish. IP strategy, you come up with a development plan, so budget, Gantt chart, medchem, and experimental protocols. Then you need a clinical and regulatory strategy, and then you actually do the new co-formation. So you license from the tech transfer office and bring together all of the other agreements and raise the capital if it's not already there. So a general framework that I've used is looking at the pharmacology or medicinal chemistry, the drug discovery aspect, which is fairly typical. What's unique is looking at the geroscience data and then the commercialization aspects, and that all comes together in translational attractiveness. So we'll dig into that. So defining a LongBio company, it sounds like a truism, but in order to be a longevity company, you need lifespan data, and that's a Kaplan-Meier survival curve. So it's pretty simple. You need to extend healthy lifespan in some model organisms, ideally mice, but I know that's hard. And it's got to be healthy lifespan extension. So for example, knocking out growth hormone doesn't count, or knocking down growth hormone receptor doesn't count because the mice are not really in great health. So if you don't have a lifespan curve, you're not yet a longevity company. So endeavor to get that if possible. In those companies that raise money in the longevity space without having longevity data are actually detracting from the overall mission. If you have a company that pivots, say, into oncology and never does any longevity work, or never label expands their drug into many different age-related indications, they're actually sucking capital away from more deserving longevity companies. So please have a plan to do lifespan and longevity studies and label expansion. So how to read these curves, you guys probably all know, but it's pretty simple. You have a median lifespan, the delta here at 50%, and then your maximum lifespan at, then LD 90 or so. And then you take a ratio of that. Anything above 10% lifespan extension in mice is pretty substantial, especially if it's healthy lifespan extension. So where to find these assets? So every VC knows you're only as good as your deal flow. So I've gotten a lot of luck with the journals. It's underrated, but just stay on top of the journals, like with Google Alerts. Going to conferences, this is good because you get the science when it's not yet been published, which can give you a year or sometimes 2 years of early bird advantage. There are also business conferences, but that's a little later stage for fundraising purposes. Tech transfer offices can be helpful, but they're very hit or miss because it's complicated with TTOs. We'll have a talk from my friend Dan Catrone in a couple of weeks about TTOs. Grant databases are quite good. Hat tip to Frank Taffy for this, and it's validated by committee at the NIH. So there was at least some quality control. Of course, inbound deal flow. Once you have your own network and reputation, a lot of deals have come to us that way. If you're a masochist, like I sometimes am, go through the patent databases. That's good because a lot of stuff is patented but not published. So there's a whole dark matter of data out there. And encouragingly, I noticed it's a wide open playing field. So for a lot of the deals I've looked at, they have not been in touch with pharma. There are just not enough pharma search and evaluation people to really saturate the world, even saturate the major hubs of, San Francisco, Boston, San Diego. Even they're not totally saturated by pharma, much less Europe or elsewhere. So here are some of my favorite resources. This is not secret. You can take a screenshot of this. Obviously, Nathan Cheng's unparalleled longevity list and market cap newsletter. This is actually really good. Fight Aging from Reason, that's a really good one as well. Leaf, Lifespan IO has a lot of good articles, longevity technology, etc. Nathan has a podcast, as well as a bunch of other podcasts sort of related. These are good news sources for biotech, some top journals. I would recommend these texts. I can actually send you PDFs if you can't get access to these. I have a whole Google Drive folder full of these books and a handful of other interesting texts. If you need those, send me a message on LinkedIn. So preclinical technical diligence, this is what I tend to focus on, where the rubber meets the road. So it doesn't matter how good your clinical strategy is, how good your IP strategy is, how good your team is, if the science is not rock solid. So everything starts with the science. And as you can see from this figure, it's pretty complicated. I mean, this is one figure from one article, and I have training in neuroscience, but it still takes some time to actually analyze. So it's difficult because you have to be conversant in a bunch of different fields and many different techniques as well. So you got to focus on the science first. If you use consultants, you got to be careful. You've got to use, you can't just use one or two, because I'll get 5 different answers from 5 different consultants. So it's really on you to drive this. Look for whether this result has been reproduced by other labs previously or similar results. Lifespan studies are rarely reproduced just because they take so long, but this is something that the field really needs, like through the NIA ITP program or a larger version of that or a private version of that. More on that later. And anyway, we're going to talk about that. So here's an example of like a structured process for finding these. This is sort of early from Apollo. I was searching for a bunch of different assets. So come up with a list, universities, and then their mechanisms of action. And then I could actually score them based on a semi-quantitative model for all of these different parameters from pharmacology and aging and IP, et cetera. So you can actually, in a pretty unbiased way, determine which are the most attractive assets. So one example, a pretty simple one, is a therapeutic index. So the ratio between the effective concentration of your drug in cells or in vivo versus the first toxic dose or the lethal dose. And you want that ratio to be as high as possible. So you can input your own numbers. Other examples we'll get into in a sec. So at Apollo, I started to create this checklist here, and it covers lifespan, aging phenotype, drug discovery, medchem pharmacology, and then aspects of the team. And then I came up with a process for actually launching a new code. You can get it done in 2 or 3 months. Usually it takes longer, but you can do it in that time period properly, I think. So we'll touch on some of these mechanisms. So first is lifespan data. This is kind of one of the key decision criteria that I look for. So you can score it based on effect size and then the model organism. So yeast, worms, flies, fish, rodents. Pretty straightforward. The more sophisticated the animal, the more points you get. Then you can look at other criteria. So, is this based on a knockdown or knockout study? Is this conditional? Is this a knockout or knockdown late in life? Have they done tissue-specific work? Has the lab identified a small molecule hit compound? Is it a peptide, antibody, or other biologic, gene and cell therapy? Has the lab done any medchem already? Is there a patent application filed in the US? Is it issued or granted? Is it issued elsewhere? Has it been through IND enabling pharm tox studies? That's pretty rare for an academic project. Is the target known? Have they done target ID or CETSA or proteomics to identify off-target binders, et cetera, et cetera? So there are a lot of different criteria you can use, and this is very simplistic one, but it gets the job done. You can also consider the people involved. So how many people are involved in this project? If we finance this work to go on at a university or with a CRO, how many people do we actually get out of it? How many people have been working on it for a long period of time? Was the key experiment reproduced, or were they independently reproduced by different people, just to make sure that it's robust? Does the PI have a lot of complaints with shoddy data to watch out for? Has the PI spun out other companies or licensed technology previously? That's useful. Is the PI focused on this project, or do they have a big lab with other fish to fry? Is it a hungry young PI, or is it a tenured, distracted full professor? That's not always a hard and fast rule, but younger in general seems to be more motivated. That's true of biotech founders too, as we'll see. What kind of papers are published? Do they have unique assays or capabilities that cannot be reproduced elsewhere, like with a CRO or a different lab? Is the TTO easy to work with? Or is the PI a compelling communicator? Will they help you pitch? That's underrated. Is there a management team in place already that's useful, or like a postdoc who's ready to spin it out? And then another consideration are the deal terms at the TTO. So upfront, usually we don't pay an upfront. Sometimes you have to reimburse patent costs, milestones on clinical development or sales, royalties on net sales, the overhead rate at that university, and who controls IP prosecution and who is an inventor. So coming into IP, IP is an art, not a science. So you want to plan for success, that your drug will be successful, and you need everything really tight, because you're going to be ready to stand up to an inter-parties review challenge once your drug is nearing the market. So the ideal situation with the patent, it's a new chemical entity with a combined composition of matter and method of use patent and a non-trivial formulation. You don't have to have all of that up front, but eventually you'd like to. Ideally, you don't want the patent filed yet. You want to wait until the last moment in order to file the patent to extend the lifespan, which is ordinarily 20 years. Sometimes you can extend it with patent term extension for when you're in the clinic. Usually, your first submission to USPTO or EPO is rejected. This is normal, so don't worry about that. It's just haggling. It's part of the bargaining process. But usually everything's not ideal. So if the patent is more than 5 years old, you can develop new analogs, formulations, salt form and crystal polymorphs, and methods of use. You want to create a thicket of patents as a deterrent to competitors more than anything else. Also note that in the US, there's a 12-month grace period on disclosure. So make sure your PI does not publish this data or present it in the conference, because if they do, it's prior art and you can't patent it. In the US, you have 12 months and you can still patent it, but in Europe, you don't. So be very careful about prior art. There's another kind of patent called a selection invention patent, which is you've patented a certain chemical structure, for example, but you find a new use for that, or you modify that slightly and it's non-obvious. And so you can refresh the IP in that way. Even without a patent, you get 5 years of marketing exclusivity from FDA, and biologics get 12 years. But if you don't have a composition patent or very strong other forms of patents, it's hard to get pharma attention. In biologics, one of the advantages of developing a biologic is they're nearly perma-patented, because it's really expensive and difficult to make a biosimilar. So Humira, the best-selling biologic, has been on the market for, I don't know, almost 20 years, and only recently have and the key patents were expired, only recently have biosimilars come onto the market. So one way you can come up with a valuation for any asset is a risk-adjusted net present value model, RNPV. This is voodoo, like all evaluations. It's not precise, but it's useful. It's a useful exercise. So as the non-finance people may not know, money in the future is worth less than money today because of discounting. So that's called the time value of money, because you can always get a return on some capital over time. Also, in addition to the discounting by time, you have to discount for the risk of failure at each clinical stage. So you can actually see here, the net present value increases to the point of FDA approval, but it doesn't go up in a straight line because you're de-risking at each clinical stage. And then once you're generating revenue, you're basically running out of patent life. So you can calculate sort of the area under the curve for what that asset is worth today. And you calculate it, your drug's particular odds of success on the basis of the therapeutic area that you're in. So oncology, for example, as you can see, just pay attention to the orange bar here. That's the total probability of success from phase 1 to approval. in oncology, it's the lowest of any therapeutic area. You'd think pharma would avoid it for that reason, but it's the opposite. Pharma, or maybe because pharma is pursuing it so intensively, it's less likely to succeed for some reason. But even though over 50% of capital goes into oncology, the odds of success are the lowest. So when you're doing this valuation model, you have to discount by that probability of success at each clinical stage. Whereas compared to vaccines, as we've seen with the COVID vaccine, that was lightning fast, because somewhere around 40% of the time, the vaccines actually get approved. They actually work. So consider your modality risk as well as your therapeutic area. And then for a lot of this, you have to meet with clinical KOLs. So here's a super simple RNPV model, back of the envelope calculation to tell you what your asset would be worth today. So simply put, you multiply your patient population with the market share, uptake rate, and price. And then that gives you your revenue. And then you take the revenue and you discount by year, and you discount by the probability of success at each clinical stage. That's your risk-adjusted revenue. And then you subtract your costs. So the cost of the clinical and preclinical development multiplied by your success in the clinic, because you may not have to pay those costs if you don't succeed in the next phase. And then you also discount by time. That's your risk-adjusted cost. And then you subtract the revenue from the costs, both risk-adjusted forms, and that gives you your RNPV today. So you can basically look at that at your leisure. And a discount rate, somewhere between 15 to 30% is typical. Right. So when you're doing company formation, these are some of the key documents here. So your development plan, your Gantt chart, what's the timeline? Pretty straightforward. And your contingency plans if something fails. And then your budget. So pretty straightforward on a monthly basis or a multi-year basis. What's your budget? So I'll give you sort of a case study of one of the companies I helped launch a couple of years ago, Samsara Therapeutics. So one of the hallmarks of aging is proteostasis failure, the turnover of the proteome. Cynthia Kenyon showed this a couple of years ago quite compellingly, that germline immortality in C. elegans is mediated by proteostasis, by the turnover of the entire proteome upon the moment of conception or egg fertilization, if you will. So this is an interesting area to be in, especially in the neuroscience space, proteostasis. So an overview of proteostasis, proteins get damaged by various things: heat shock, regular metabolism from oxidative stress, ER stress, if the protein is not folded properly, whatever. Protein gets damaged in various ways. It can be degraded by the proteasome if it's ubiquitinated. There are a couple companies working on this in stealth mode, enhancing proteasome function. Chaperones, these little Pac-Men here, can refold it. Heat shock factor governs a lot of them, and one reason by going in the sauna and being exposed to heat might actually be beneficial because it turns on heat shock factor. If the proteins just aggregate and gum up the works, it causes aging. And then finally, I think most proteins actually are degraded by autophagy, either macroautophagy, not shown here, microautophagy that Kenyon pointed out in that paper, as well as chaperone-mediated autophagy. Anna Maria Cuervo is working on this. But chaperone-mediated autophagy only deals with about 30 or 40% of the entire proteome, whereas macroautophagy can break down pretty much everything, not just proteins, but whole organelles as well. You name it, anything within the cell pretty much can be broken down by autophagy. So what is autophagy? It comes from the Greek self-eating. There's a Nobel Prize award a couple of years ago for this work. Basically, an isolation membrane buds off from, say, the endoplasmic reticulum. It encircles, with the help of cargo receptors, whatever needs to be degraded. Maybe it's the peroxisome, or maybe it's the mitochondria. Forms this double layered vesicle, the autophagosome. Autophagosome has to fuse with the lysosome, which contains hydrolases, these enzymes, as well as a lot of protons, a lot of acid to break things down. It's like the stomach. It's like the stomach of the cell, basically. And it turns out that autophagy is probably really important for the brain, also the immune system. So the rate of autophagy, the autophagic flux, declines with age in humans and in animals, other animals. And interestingly, this is kind of an aha moment for me. When you do a whole body knockout of ATG7, the mice died because of neurodegeneration first, followed by bacterial infection. So if you knock out autophagy in the whole body, you could expect the system to break in any number of ways, but actually neurodegeneration is the weakest link. So that indicates that the CNS is most heavily dependent on autophagy for its function. Knockout of autophagy during development is lethal, but conditional knockout results in neurodegeneration, suggesting that proteostasis failure is critical in neurons. It could be because neurons are kind of like the supercentenarians of cell types. Neurons, at least most neurons, are with you for life. You have them by the time you're a kid, and they need to survive for your whole life. Unlike most other cells, like the liver, right? You can cut off a third of the liver and it'll regenerate. That's not true of neurons. So neurons have to invest in all of these self-repair and longevity mechanisms, and proteostasis is one of them. It's also the first line of defense against intracellular pathogens, any pathogen that gets inside the cell, from viruses to bacteria, et cetera. And actually neuroinflammation is increasingly driven by microbes in the leaky blood-brain barrier. So at Samsara, we actually have data, I can't say too much, but we have data in many interesting pathogens in terms of clearance with these new mechanism of action autophagy enhancers. And that's interesting because it's a host-directed therapy, so microbes are unlikely to be able to develop resistance from it. And it works against many different types of microbes too, kind of like fasting. So autophagy, you can basically clear out the junk that accumulates with age. There's a Nobel Prize awarded. You can clear many different molecules. Fasting and exercise are drivers of autophagy. And actually, for animals to derive the benefits of fasting and exercise, autophagy must be genetically intact. If you knock out or down autophagy, you don't derive as many benefits from fasting and exercise. So also, genetically enhancing autophagy is sufficient to extend healthy lifespan. The mice don't get fat if you overfeed them. They're more muscular and physically active. They're younger in every way that you look. And a couple of different groups have shown that already. So Samsara, this is a platform company developing autophagy enhancing small molecules, raised about 15 million to date. And we have over 4 small molecules that are new mechanism of action, independent of mTOR, that enhance autophagy. So this is some of the team. I founded it as the EIR at Apollo. I pulled together the team and the business plan and everything else. And we brought on a number of rock stars from biotech and venture, as well as our scientific founders, Guido Kromer, who was the senior author on the Hallmarks of Aging paper that you'll know, and Frank Matteo, his longtime collaborator, who's a great yeast geneticist, figuring out the mechanisms underlying these drug activity. And then a handful of other great scientists in the team, and the team is growing. So reach out to Samsara if you want a job in the UK. So we've gotten a fair amount of press coverage. One is Financial Times voted best pitch for the company at the Financial Times conference, and then a bunch of publications as well. So we're not the only ones in the autophagy space. We're going after different mechanisms of action than pretty much everybody else, though. So Self-AG, Life Bioscience-backed company, is pursuing chaperone-mediated autophagy. Calporta was recently acquired by Merck for up to $576 million. In the CNS, Casma Therapeutics, launched by my friend Tim Funnell, by Third Rock, Caraway, and Libra. And there will be undoubtedly more. The problem is they're mostly focusing on lysosomal targets like TRPM. And so they're overly concentrated on one target class, but we'll get into that another time. All right. And ironically, because I have a cold right now, I wish I could have benefited from this therapies we're working on at Immuneh. But the next company that I'm launching is called Immuneh Pharma, and we're targeting the bone marrow in particular, as well as circulating immune cells. And so just in summary, as a teaser, we're still in stealth, but we're working on small molecules targeting HSCs ex vivo and in vivo. One of our compounds has shown 10 to 15% median lifespan extension in age wild-type mice, even when you dose for 1 week only in old mice already, 75 weeks of age. This is a big deal. I mean, rapamycin is the only molecule that comes close when you start giving it late in life. And with rapamycin, you have to give it chronically. Whereas with this compound, you only have to give it for 1 week, and that's not optimized. We might even get increased lifespan extension if we give a larger dose or a different dosage regimen. The drug also resets the epigenetic clock by flipping an epigenetic switch in stem cells, including the hematopoietic stem cells, to reboot the immune system. It's not related to any known hallmark of aging that's been drugged previously, such as telomeres, sirtuins, autophagy, et cetera, or senescent cells. We can dose HSCs ex vivo with this compound. It's a negative allosteric modulator of a known but hard to drug target, but the team has managed to drug it. It reverses the myeloid skewing with age, restores youthful immune function, response to vaccines, and reduces the common pro-inflammatory cytokines that drive immune inflammation. Founders and collaborators in US, Germany, and Switzerland were looking for more. We want to build a pipeline of assets around immune rejuvenation as a general theme, focusing on ex vivo incubation of drugs, such as HSCs, T cells, and NK cells to turbocharge them, because a lot of patients cannot benefit from these new exciting therapies because their cells don't really work ex vivo because they're too old. So we're hiring people with backgrounds in immunology, pharmacology, medicinal chemistry, drug discovery, and remote work as possible. So send me a message on LinkedIn. All right, finally, some closing thoughts and sermonizing here. So as a call to arms to the field, you're only as old as your arteries, said Sir Thomas Sydenham, an Oxford physician in 1600. Cardiovascular disease kills 60% of us. It's not enough to work on new mechanisms. There's not enough work on new mechanisms in CVD. So most money goes into oncology, even though oncology is not the leading cause of death. It's heart attack and stroke by far. Animal models are unreliable, like these Alzheimer's models and Alzheimer's that have led us astray for 20 years plus. If you can slow aging in wild type animals, preferably mice, that's a more robust and less contrived intervention that I think is going to work across many other indications. Because aging itself is conserved across species remarkably closely. So we really need a mouse aging CRO. So when you want to buy pre-aged mice, JAX is expensive and often sold out. NIA doesn't have as much, and we need different genetic backgrounds like the Het3 mice that the NIA ITP program and Richard Miller use. So please, somebody out there, make a pre-aged mouse CRO that does lifespan studies. The whole field is bottlenecked by this, and you can basically copy the playbook from the NIA ITP, and hat tip to Kristen Fortney at BioAge for echoing this point. This is a big one. So there's also been a renaissance in natural products, drug discovery, and phenotypic screening. So almost all geroprotectors, from rapamycin to metformin to fisetin to you name it, are natural products or derived thereof. I don't think that's an accident. And a lot of these compounds are coming from phenotypic screening as well. And phenotypic screening has advanced with new target identification methods such as chemoproteomics. So this is back to the basics of the last century of what pharma did in their golden age before R&D productivity began to wane. So why is productivity waning? As Jacques Scannell, a thinker, coined the term E-Room's law, it's the inverse of Moore's law. It's getting exponentially more expensive and time consuming to get a new drug approved. There are a lot of factors involved here. Some of them, one is pharma consolidation. They're fat, happy, incompetent, and they're cartelized, so they don't need to innovate. Pharma spends a lot more on marketing than on R&D, and spends more on paying dividends and share buybacks than they do on R&D. So that's actually a problem, because they're sucking capital out of the biomedical system and paying it out to shareholders or spending it on marketing. The other argument is that the low-hanging fruit has been picked, so the easy diseases are cured or well managed. I don't really buy that one. The regulators are getting more strict. They're certainly not in oncology. I don't know if they really are getting that much more strict in most areas. There are some fundamental assumptions about drug discovery that are wrong. A Faustian bargain made with alluring new technology. So we'll get into that in a sec. I think that's one of the major ones. The other one is that the pharma lobby is cooking the book. So the real cost per approval put forth by groups like Joe Damasi at Tufts are exceedingly high. And there are a lot of articles in various journals critiquing these estimates of costs $1 billion or $2 billion to get a drug approved. Well, yeah, that's just because you're wasting most of that money in the process, but it doesn't actually cost that much to get a drug through clinical development, maybe 1/10 of that for a large indication. So they're overinflating their costs in order to justify high drug prices. I think that is true, but it's probably not the whole story either. So dealing with the history of drug discovery and the modalities across time. So for thousands of years, we just had plants pretty much, botanicals. Some are known here. Some of the most important drugs, you know, the opioid painkillers, some of the cardiac glycosides, aspirin, etc. These have been the bread and butter for a long time. And then around the 1850s, you know, coming out of the chemical dyes industry in Germany, the pharmaceutical industry was formed, and they started doing chemistry on synthetic dyes like methylene blue and Paul Ehrlich, as well as modified natural products. And so, you know, coming into the 20th century, this is kind of the golden age of pharma and medicinal chemistry. Most of these compounds were actually analogs of natural products. So the statins, or metformin, or everolimus, which is a rapamycin analog. Many of these compounds are actually derived from natural sources, or from, say, neurotransmitters or whatever. And there's a really interesting story about the Nobel Prize that was awarded for artemisinin, which is the key standard of care molecule for malaria, which saved many millions of lives. And then coming into the 80s, we had a sort of sea change where we had the molecular biology, biochemical revolution. So you could start to screen drugs in a dish against a single protein, basically. And you could do large-scale chemical synthesis like combinatorial chemistry. So we had these huge libraries, started to screen them against compounds or proteins in a dish. And it actually didn't yield that much, except for in certain very narrow domains. And now there's sort of a renaissance going back to phenotypic screening. Some of the problems with CombiChem have been solved with DNA encoded libraries, but I think we're sort of marrying those two areas of chemistry and drug discovery. And then in parallel, you've had the dawn of biologics, monoclonal antibodies, and now more recently gene and cell therapy. And I'm very bullish on the long term for gene and cell therapy, but in the next 20 years, it's going to be really tough. I just want to give a shout out to high content imaging and phenotypic screening, CRISPR screening, and iPSCs, as well as chemoproteomics for target ID. So when you do a phenotypic screen, you're looking for a phenotype in the cells like autophagy enhancement, but you don't know what the target or targets of that molecule are. So you need to find them. You need to pull them out. You can do, you know, affinity-based labeling, or you can do label-free chemoproteomics. And so there's been a renaissance in that. So I'm very excited about that as well. Don't really have time for this, but long story short, there are certain advantages to natural products chemistry over synthetic libraries, especially in the aging space. And about 75%, a friend of mine, David Newman, did this analysis at the NIH, finding that 75% of oncology drugs were natural products or natural product derived, even though pharma mostly screens synthetic libraries. And then a lot of drugs are derived from phenotypic screening, even though very few groups are doing phenotypic screening. So it's punching massively above its weight for reasons we can get into later. And a new development that I like is Scripps Research Institute, which is the best private drug discovery institute in the world, though I'm biased, is getting behind phenotypic screening in a big way. So I'd recommend this BioCentury article. They have great chemists, they have great, you know, high content imaging and great chemoproteomics capabilities. So keep an eye out for assets coming from Scripps, and they have a whole aging sort of area now as well. So, but not before I can out-license them. All right, so I'll leave you with this. This is the last real slide. So as a word of encouragement, most successful biotech founders are younger than you'd expect. So Bay Bridge Bio, which does a lot of great analyses, actually looked at companies in the last 50 years with market caps over 10 billion. And they found this histogram of age at founding of the founders. So for Gilead, Michael Reardon was 29, Bob Swanson at Kleiner Perkins and Genentech was 29. You know, pretty young, not the gray hair that you usually see in big pharma. Those are people who are sort of plugging away, but for some reason, younger people tend to launch more newcos, or maybe they're more intrinsically successful. But in any case, a word of encouragement that actually young people have the energy or drive, or naive enough to actually launch a new company and be ambitious. And I'd really recommend the book Genentech, The Beginnings of Biotech, for sort of a colorful account of what the groovy days of biotech founding were like. So with that, thanks for listening. I'm happy to answer any questions, and I want to give a shout out to my friend Nathan Chang's new fund, which I advise, HealthSpan Capital. If you'd like to invest, they take relatively small check sizes, and we want to invest broadly in many different Geroscience LongBio companies. So ideally, everybody in the cohort, with a good idea, we are interested in investing in you too. So anyway, I'll leave it at that, and I'm happy to take any questions.
Great, thank you so much, Sebastian. That was great, amazing, and yeah, I really enjoyed all you had to say about giving us a little bit of the background, the bigger overview picture of where Geroscience and LongBio fits in, and some of the checklists that you had were really insightful and great tools. I definitely encourage everybody to rewatch that part of the presentation. But before we open up for audience Q&A, actually, we're a bit short on time. Do you have a couple, maybe 10?
Yeah, sure.
We can go a little over. Okay, awesome. Thanks, Sebastian. So before we open up, are there any other things that we could do to help you? Spirit of service here on deck. I know you mentioned you're hiring. Maybe you could say a couple of words just to reiterate about certain roles that you're looking for.
Yeah, sure. People with immunology expertise, as well as drug discovery and operational background. So, and even if you don't have that much drug discovery expertise, but you're just very competent, conscientious, organized, and intelligent, I want to talk to you for this company and for others. I work with a handful of different groups that are looking to hire people in the virtual biotech capacity. So yeah, just send me a message on LinkedIn. Let me know you're interested in being in the Rolodex. And then we're not currently raising money for Immuneh right now, but within about a year or so, we will be. So if you're an investor and you would like to be there for the Series A round, just send me a message and we will follow up and keep you posted on our progress once we're out of stealth mode.
Okay, awesome. Excellent. Okay, so let's open up to some audience questions. Hanadi, do you have a question?
I do. First, Sebastian, that was such an amazing talk. It was really informative. And personally, I do owe you a follow-up to actually get into diligence with Juven. And after this talk, I'm actually a little nervous about that. But
no, Juven looks great.
So one question I kind of had was around sort of your requirement about companies really performing lifespan studies. So I really feel like it really depends on the therapeutic modality. So for example, right at Juven, we're really developing protein-based therapeutics that can promote tissue regeneration, where a health lifespan study in mice might cost, just for a single study, over $1 million. And there still might be nuances where, even if it's a mouse protein that we're using instead of the human, if it's produced in, for example, E. coli, it could still cause an immune response over a long period of administration. And using simpler kind of organisms don't exactly recapitulate some of the mechanisms of action that we're really targeting that are more really related to mammalian aging. And so, why is that like a very hard requirement in your opinion? Or do you think, companies that are also really focused on, enhancing regeneration in the age context, is it really necessary to really perform lifespan if your actual modality causes some either cost prohibitions or other issues as mentioned, like immune response based on the testing?
Yeah, that's a fair point. Lifespan studies are time consuming and fairly expensive. They can be done for below 250K with 50 to 100 mice in the right places. But yeah, I mean, if you need to produce a protein and it needs to be very pure and manufactured in mammalian cells, that's going to be expensive. So I mean, I would understand if you hadn't done that, but to really attract a lot of longevity interest, it's always good to have longevity data per se. I've invested in a company previously that didn't have longevity data, but did have pretty amazing tissue regeneration data. And so obviously I'm not a complete ideological purist there. Tissue regeneration is cool, but then it would really be better classified or branded as regenerative medicine company rather than a longevity company, because by definition, longevity requires lifespan extension, at least healthy lifespan extension. And so showing that across the lifespan is kind of sine qua non. but yeah, I mean, I still think there's plenty of room for companies that have not yet done lifespan studies, but it would be great if there were some plan, some budget to do lifespan studies, or at least in aged animals to show extension of functional healthy lifespan, if not lifespan per se.
Absolutely. I totally agree. Sebastian, thanks for clarifying that.
Okay,
Cheers.
great. Thanks for the question, Hanadi.
Mhm.
Okay, so we have a question in the chat from Niklas Rintdorf. With regards to aging as an indication, what is the success rate you are using in your models from start to finish? So like the 10% average by Andrew Lowe, or yeah, just curious what you think about the success rate for, I guess, Gero.
Yeah, it's sure. That's kind of a quick trick question because it's like dividing by zero, right? It's never been done. The closest that you can get are like preventative studies like metformin or statins, and the effect sizes with metformin and statins are very weak. So we shouldn't hold those aloft as the standard. I actually think metformin is a little bit of a red herring because the lifespan extension in mice is only 5%. And it's got to be the perfect dose level too. So I think that's actually possibly a mistake to do the first really big longevity study with metformin, but it's safer than rapamycin, so fair enough. But maybe fisetin would be a better option. Anyway, so there is no approvable indication, no label for longevity, and such a study would take too long unless you do the sort of morbidity latency type model from Barzilai et al. So we don't try to model that. All we model is the lead indication and then maybe 2 or 3 label expansion potentials. So for example, if you have a compound that rejuvenates the hematopoietic stem cells, you could go after various types of autologous bone marrow transplant, like multiple myeloma, for example. And you could also go after certain cell therapies that are being derived from hematopoietic stem cells. Then you could look at in vivo dosing for rejuvenating the immune system in a vaccine enhancing setting, which Bio tried to do with their rapalog, which I hesitate to even call a rapalog because it was a general kinase inhibitor, right? It hit PI3 kinase. It was just kind of like a compound that Novartis had sitting on the shelf. With all due respect to my friends at the Bio team, that may not have been the best compound to carry forward in an aging study like that. Anyway, so you can just model multiple indications, and if it's successful in at least one of those, it's going to be, sufficiently profitable to be interesting to pharma. So we don't even account for the longevity aspect. We've got to wait another probably 10 or 20 years before we can start to include that like general prophylactic dosing. But then the market is so big, it's not even worth calculating. And this is one of the things we've got to get across to potential investors is there's this whole dark matter, iceberg beneath the surface of if you have a really safe drug that can be chronically used to slow aging, then the market is massive for that. And it's just not included in any of the calculations. So anyway, yeah, we don't model longevity as an indication in the RNPV models, but hopefully one day we will.
Okay, awesome. So next question, we have Brad English. Would you like to come up and unmute?
Yeah, sure. Thanks for the talk, Sebastian. I was just wondering, well, just to give you back the floor, because you kind of hinted at it earlier, if you could speculate further on why you think there aren't many DNA damage repair interventions currently being attempted. Thanks.
Sure. Yeah, good question. Well, there was at least one. There are a couple companies out there doing it, but they're focusing on transposons, which is not really DNA repair, but it's just preventing genomic instability, or sirtuin 6, activating sirtuin 6, for example, Vera Gorbanova's group, which is a good one, and some others. There was a group from Jan Hoijmakers, one of the great figures in the field in Holland, that was called DNAge, I believe it was called, and it was backed by LSP. And the initial intent was to enhance DNA repair, but the investors, as often is the case, pivoted them to something easier, which was not enhancing DNA repair, but inhibiting DNA repair, like, for example, the PARP inhibitors do. And that'll work in cancer, but it'll cause more problems down the line, just like chemotherapy in general. Chemotherapy and radiation cause DNA damage, and they cause accelerated aging. So one of the reasons is there's no clear pathway or precedent for a DNA repair enhancing drug. You could, and it's not clear what the indication would be either, because, there are many diseases that are worsened or caused by the slow accumulation of DNA damage, be it cancer or neurodegeneration, just slow, continual hits of DNA damage. But you'd have to prophylactically take that drug for a long time. So it'd have to be a long study. The alternative would be progerias, or diseases like Li-Fraumenti syndrome, which is a loss of function P53, puts you at risk of cancer. So it's just not clear what the indications would be. And those are relatively rare indications. Of the progerias, there are only there are only a couple hundred or less than 1,000 people with Hutchinson-Gilford in the world, or Werner syndrome in the world. So it's really not clear to me how you would proceed from a clinical indication standpoint, and you'd have to do long prophylactic preventative clinical trials. So that could be part of the reason. And also another reason is DNA repair is super complex. I mean, you've got all these moving parts. I don't know if you've seen the videos, but it's not just like you need to inhibit an enzyme. You need to activate a whole set of proteins that work together and do kind of elaborate manipulations of the DNA. So it's hard to find a drug that does that. But the way around that would be, all right, let's find a drug that works upstream of the actual DNA repair machinery itself, but recognizes DNA damage, like sirtuin 6, for example, or like the PARPs. And you could argue that NAD kind of does that. Could argue that higher NAD levels activate PARP, and then you're going to recognize more DNA damage and therefore repair it before it becomes a mutation per se. So there are ways around it, and there are groups trying to suppress transposon activity, the jumping genes too, but that's not really DNA repair. So there's a little bit, but I'd love to see more. And someone who could just come up with an assay for DNA repair and just do a phenotypic screen for molecules that enhance DNA repair, that would be a pretty cool project, I think. Anyway, probably more than you asked for, but hopefully.
No, that was a wonderful resource of information, and we might have someone might have something like that coming down the pipeline. But thanks a lot for your information.
Nice. All right, keep me posted. Cheers.
Great. Thanks, Brett. Actually, I have a question. So, Sebastian, when you're doing like a preclinical diligence, or diligencing the science, typically how long do you take to go over one idea? Just roughly the timelines for that, and the kinds of people that how many different people will you reach out to do sort of like an advisory or something like that?
Yeah, it really varies a lot. Sometimes, I mean, if we're writing a big check, it's proportional to the check size, really. And it's also proportional to like the team quality. If it's a team that doesn't have that much expertise, or they haven't explained it that well, we've got to do a lot more legwork figuring out if it's legit. Whereas if it's, everything else is very well done and organized, then we don't really have to double check as much. So I'll usually probably, for any given project, if it's a large check size, spend probably about a week, not a week straight, but a week on and off, looking into the literature, calling up some KOLs, sending back and forth some questions. Sometimes that takes a couple weeks of diligence. So usually I like to come to a go, no go decision within 2 to 4 weeks on anything that comes across my desk.
Wait, awesome, great, thank you. So next question, we have Kyle Brewer. You can come off mute, thanks.
Hi, Sebastian. Thanks for talking about the usage of natural products. There's a couple of companies who have been successful with that, like, excuse me, Elysium and Chromadex. If those companies were starting today, how would you value them, and what would you do differently?
Yeah, the Elysium Chromadex saga is quite a messy one. I don't know if you know the backstory there. They're supplement companies, so it's a little bit different to model. You're modeling revenues and so on. And Elysium does have pterostilbene in their mix, which is a natural product in blueberries, which I take and I used to work with in the lab, which is just an O-methylated version of resveratrol, which is more potent. But NR, I don't think NR is a natural product per se, but it's like an analog. Actually, no, NR is in milk, so it is a natural product. But it's an analog of niacin. And I think the jury is out on how much better these NAD precursors are relative to just high dose niacin. But we have seen some interesting data, for example, with NR in hematopoietic stem cell function. But the bioavailability of the NAD precursors is pretty low. It seems to be metabolized into niacin pretty quickly, so you don't get high levels in cells. But working on new analogs that slot into a different part of the NAD salvage pathway may be worth pursuing, looking into that now. As for how to value those companies, really hard to say. Fortunately, you can value them in a simpler way today, which is just their some multiple on revenue. But then you have to account in both of their cases for litigation risk. They're both suing each other over who invented what first, or who's stiffing who on the supply, and who's making what in China rather than where they should be making it, et cetera. So yeah, I don't know. I haven't kept up with that whole saga as much recently, but I'm in favor of supplements. I mean, in principle, a molecule is a molecule. I mean, supplements are great. I take a ton of supplements. They can be supplements because they're safe. Just because something is supplement doesn't mean it doesn't work. It just means nobody has really bothered to do the rigorous clinical trials that are expected of drugs, because drugs tend to be toxic. So if you could do a supplement and have a rigorous trial and then get that supplement approved, you would get at least 5 years of marketing exclusivity. And there have been cases of that. Botanicals approved, like the catechins, the polyphenols from green tea, for example, are an FDA approved drug. And there are a couple of those in Europe, has a bunch of approved botanicals too. So they're just very safe molecules that often haven't gone through the same rigorous clinical development, but they should. There's no reason they shouldn't if you can get an IP position. Anyway.
Okay, great.
Fantastic. Thank you. Great answer.
Thanks for your question, Ka. All right, moving right along. Aaron, can you come off mute and ask a question?
Hey, so for Samsara, how do they measure autophagy? And were they were just doing it in mice, or did they also do it in humans?
Sure, there are a bunch of ways to measure autophagy. One way is the ratio of LC3-1 to LC3-2, so the lipidated form of this sort of cargo receptor LC3, which is present on the autophagosome. That's a sort of Western blot-based assay. You can also do a stable cell line of the traffic light reporter, it's called. So you have GFP and RFP conjugated to LC3. And as autophagic flux progresses, it quenches the GFP. So you go from yellow to red. And you can do that in high throughput screening. We don't have data in humans yet. That would be risky, although maybe one of our gung-ho scientists would test it on their own blood or something ex vivo. But there are data of other autophagy enhancers in human cells from.
Those are
the
methods that Samsara used.
Sorry.
That's what Samsara did, was those two, the LC3 and the GFP version.
Yeah, those are some of them. We have a couple more related to lysosome function. Some of that is kind of secret sauce.
Okay.
And in addition to the lysosome function assays, it's a platform called LysoTracker. One of the cool things that Samsara has developed, independent of me, is a target ID platform. So basically a way of doing thermal proteome profiling to assess basically whole proteome target engagement of your small molecule. So it's a target ID method that the team has made more efficient. So we can do phenotypic screening and then pull out the targets relatively rapidly. So.
Okay. How large of a breakthrough would it be for there to be an in vivo, near real-time way of measuring autophagy in the human body?
Well, in the human body, it would be fantastic. I mean, everyone's looking for a biomarker of autophagic flux, and some groups are looking at metabolomics. But if you have a better idea, I'm all ears. We'd love to have that, especially as we go into the clinic, just to assure target engagement. Yeah.
Great. Yeah, that's
So, That's tantalizing. You got to send me a message on LinkedIn, man. I want to hear more.
Oh, no. No, I know it's important. I don't know how it's would be done. Nobody really does, but I know that that's like, there could be a massive breakthrough there. So
You can do it in peripheral blood, but you know, autophagic flux in blood cells doesn't necessarily correlate with your brain or tissue of interest.
Yeah.
You know?
Okay, great.
Cool.
Thanks for your question, Aaron. Nicholas, can you come off mute and you can ask a question. Thanks.
Yeah, thank you. I have one question regarding your thoughts on phenotypic screening. There are basically these two camps, and I wanted to know where you locate here. You have this sort of generalist phenotypic profiling camp. They do cell painting. They analyze the whole organism with a whole cell or an organoid to look for differences between aged and young cells, for example. And you have this sort of more process-specific camp that's thinking of, you know, for example, autophagy, and is then implementing lyso tracker staining routines to look for the sort of drugs that engage that particular mechanism that they're interested in. Now, there are companies that are pretty successful with both of these technologies. Where do you sort of locate yourself when it comes to aging as a drug indication for phenotypic drug discovery?
So there are groups doing the sort of unbiased approach of taking pictures of old cells and then giving them a drug and seeing if they look younger in a way that is not perceptible by the human eye, but is perceptible by the machine. That sounds cool to me. It's one of those hypothesis-free approaches. It hasn't, to my knowledge, revealed anything that we didn't already know about, but it's still early. So we may find something that way. So the jury's out on that. As for phenotypic screening for a specific biochemical or molecular process like autophagy or like senolysis or telomere elongation or, you know, or even at the target level, sirtuin activation, something like that, or the aged epigenome, those are a little bit more specific and probably a little bit less of a black box. So I've seen most efforts approaching those aspects, but you know, I think they're both compatible. So if a group, I mean, someone's got to set up a CRO to do lifespan studies. There are some worm lifespan studies. I don't think there are any fly lifespan groups. There may be one starting in Denmark. And then no mice other than jacks. So in addition to that, you could also do cell-based aging if you had, and you could also couple it with a bunch of age-related assays, epigenetic clock, as well as the machine learning-based cell age pictures. So yeah, someone, you know, can put that together. There's going to be demand for it. So yeah.
Great. Awesome. Thanks for your question, Nicholas. I think we have one more question if you have time. I think you have time for his question. Avi, nice to see you.
Oh God, here we go.
You know what I'm going to ask?
Yeah, man, go ahead.
Yeah. Hey, Seb, tell how do you stay this young? You already hinted at the at your massive protocol. You wanna close this talk with your protocol?
For sure, man. And you've been using the protocol too, I'm well aware. So in addition to all the supplements and all of the stuff that your grandmother told you to do, the real secret, and they don't want you to know this, is actually human breast milk. You know, NR is in the human breast milk. I get mine delivered by Craigslist every morning, fresh. That's the true secret. So now you know, and then get a blood boy, as described in that excellent TV show talking about the Peter Thiel rumor. You got to get a young blood boy. That's the key. So if you're looking for one, I know a guy who can hook you up. Just reach out. And actually, in all seriousness, for those who don't know, it seems that it's not really that there's that much good stuff in young blood. It may be that it's more that there's bad stuff in old blood, which is in a sense convenient because you can just scrub out like dialysis or anapheresis, the bad stuff, or you can neutralize them with an antibody. So actually young blood may not actually do that much. So your young people are safe for the moment. but the parabiosis work is interesting and something that I've been wondering. I don't know if anyone's done a lifespan study with parabiosis factors. I can understand you wouldn't do it with true heterochronic parabiosis because they're surgically linked, the animals are not happy, et cetera. But just the repeated injection of young blood. I know that some of this work was going on in the Convoy group at Berkeley and elsewhere, but I don't
Sebastian,
recall seeing lifespan. Yeah.
Harold Catcher is doing a lifespan study in rat funded by he in Belgium, Didier Cornell and others. So, and they have released a little bit of preliminary information about that, but it's a lifespan study.
Excellent. Yeah, it's about time someone did that. I think I did hear about that. And so, yeah, I'm all for it. It's about time somebody did that.
Okay, great. Thanks for your question, Avi. I guess that wraps it up. If anybody wants to connect with you, Sebastian, where can they find you?
LinkedIn is best. Just send me a message with your ask and happy to chat.
Okay, awesome. Well, thanks again for the talk. It was amazing and awesome questions from the audience. I guess hopefully we'll see you again at some point, maybe in the future. And thanks again for all your support for the ODLB community. It's been great. Thanks, Sebastian.
For sure. Thanks, man. Happy to.
Okay, great. Awesome. Take care. See everyone.
Bye.